Hi, my name is Tychele Turner, and I'm an assistant professor at the Washington University School of Medicine. Today, I'm going to be talking about the application of long-read sequencing in human genetics. In my lab, we're very interested in using genomics to solve both rare and common disease. And in particular, we currently focus on autism and other neurodevelopmental disorders, 9p- syndrome, and undiagnosed disease. Here, I'm showing you a typical genomic workflow going from the patient presentation all the way to variants and interpretation. I want to point you to a specific part of this workflow, though, and that is the sequencing step. So currently, the state of the art for large-scale assessment of many genomes is to do short-read sequencing on a sequencer like the one shown here. And in my previous work, and also in present work, I focused on sequencing tens of thousands of individuals and looking at that data. Now, we are kind of moving into a new era in human genetics, and it's a very exciting time. And it's brought about by this new technology called long-read sequencing. And in my lab, we've had the opportunity now to look at 78 individuals using this technology, and that's the main focus of my talk today. So here's the overall outline for my talk. First, I want to talk to you about genomic variation and technology. As you may be aware, there are many types of variation present in the genome. It can be small variation, like single nucleotide variants, so those are single nucleotide changes. Can be small variation in small insertions or small deletions, can be multinucleotide variant changes, and there can be STR expansions as shown on the top right. We also have medium to large variation, including things like duplications, deletions, inversions, and translocations, where a piece of another chromosome can move to a different chromosome, as shown in the bottom right. Now, over the years, many genomic technologies have come online, and each of them is really good at doing certain things. And so I'm showing you here a table that kind of summarizes what these technologies can do. And what I hope you can appreciate is that on the far right, long-read whole genome sequencing is able to do many of the different detections for these variant classes. I want to make a point here that throughout the remainder of my talk, when I refer to long-read sequencing, I'm referring to PacBio HiFi long-read sequencing with high accuracy and approximately 20 kilobase pair reads. So if we look at this table for a few seconds here, what you can see is that things like single nucleotide variants, small insertions, small deletions, they're not detectable in karyotypes. They are detectable on SNP microarrays and in whole exome sequencing data in certain locations, and also in short-read whole genome sequencing data in regions where we have high mapability. Now we can detect these variant classes in long-read whole genome sequencing data as well. Multinucleotide variants are somewhat similar. STR expansions are really well detected in long-read data, but there's been a lot of bioinformatic work to really improve this in short-read whole genome data. So the bottom half of the table focuses on structural variants and copy number variants, and you can see that until recently with the long-read sequencing, it's been very hard to detect that kind of intermediate size structural variation, so between 50 base pairs and 1,000 base pairs. And moving along the rest of the table, you can see that some of the bigger events have been detectable in other technologies, but also detectable in this new long-read data. So now that I have given you hopefully a sufficient background, I want to move into long-read sequencing itself. So we received our first long-read sequencing genome in December of 2020, so about 5 months ago. And as soon as we got that data, the first thing that we worked on was an analytical workflow. And this is the analytical workflow that we currently use and have developed in the last few months. Importantly, I want to focus at the top on the utilization of both case and control data, and starting with the raw PacBio sequencing data files. We find this to be very critical, and I'll talk a little bit more about the controls in just a moment. If we look at the left side of this diagram, though, we can see that there is an assembly-based approach aspect of this workflow. On the right side, we see there's a reference-based approach. So the reference-based approach is actually very similar to what we've done previously with short-read sequencing data, just some of the tools are a little different, but they're the same idea. So on the reference-based side, we used a mapper called PBMM2 to align our reads to a reference genome. We use most depth to build read depth profiles and PBSV to call structural variants. We've built a custom workflow that can integrate these two data sets into a combined copy number and structural variant data for each individual. And then we can combine that data using Survivor to get a final joint call set of all samples. On the far right, you'll see what we use for single nucleotide variants and small insertions and deletions. So we first call these with deep variant. We then phase them with WhatsApp. And then we take all of the samples, all this information, and we joint genotype with the GLNexus joint genotyper to get a final combined data set. So all in all, we have a very comprehensive workflow that can detect many forms of variation, if not all. And it's been a pretty fun time. I do want to note, though, that there's a lot of future development. I mean, we're working on some things and others as well. So I think this is only the beginning, and it's a very exciting time. The first piece of data that I wanted to show you was actually for single nucleotide variants and indels, because it was quite surprising to me because newer PacBio HiFi data is highly accurate. And this is pretty amazing. So here I'm showing data from an individual who was sequenced by Illumina and the same individual sequenced by PacBio HiFi and run through the deep variant workflow. So it's the exact same kind of analysis, but just different data input. What I hope you can appreciate on the left-hand side of the slide is that generally the distribution of different variant types is the same. In the middle, you can see the TI-TV ratio is good for both. On the right, I think this really shows the main benefit in this size range for PacBio, and that is that beyond being able to do single nucleotide variants, we can now start to look at indels in a better way. So on the top, you can see that with the short read data, we can find indels between about a 50 base pair deletion to about a 40 base pair insertion. If you look along the bottom, though, with PacBio, we can reliably get insertions out to about 500 base pairs. So this was really cool. And the first individual that we sequenced and looked at, we saw there was this novel variation. So there's a repeat expansion detected in this individual, and that's shown with the little box on the bottom right of this T-view plot. This individual has autism, and this repeat expansion happens in the 5' UTR of the FMR1 gene. Now, by a pathogenicity scoring software, this was indicated as an important variant, and it was not present in control variant databases. So we were quite excited. However, being a human geneticist, I was a bit skeptical because that variation does not look like what you would normally see for an expansion in someone with Fragile X syndrome, for example. It looks too short. So that led me to the next area, which is looking at public data sets to generate frequency assessments. Now, as I mentioned, no public data had seen this variant. And so what we did was we looked all over on the web trying to find public PacBio HiFi data. And we identified 49 PacBio HiFi genomes from a project called the Pan-Genome Project. And we accessed this raw data in January 2021, and we processed all of the individuals from the raw data through our bioinformatic workflow that I showed you a few slides ago. When we did this, to our great surprise and somewhat to our relief, this variation has an allele frequency of 27.6% in these pan-genome individuals, which indicates that it's probably not relevant for our patient, but it told us that having control data was very important for the type of work that we're doing when we're looking at patients. The other cool part about the pan-genome data is that you can do things that actually I wasn't quite sure we would be able to do with PacBio HiFi data. We can actually, because it is so accurate, we can do the types of things we used to do with Illumina data, and pretty much we still do with Illumina data. But one thing that we can do is what I'm showing here, is a principal component analysis. And this shows individuals from different populations in pan-genome, and also shows individuals we have assessed in my lab. So if I break this out so you can see it better, what we find is that the PCA actually splits people out by ancestry pretty much the way we would expect. And so on the left-hand side, you can see different populations in Africa. Moving to the middle, you can see that there are people from Puerto Rico, and then moving to the right, you can see individuals who are from the Americas and East Asia, South Asia, and Europe. And as you can probably notice on the right-hand side, ours are labeled in little squares with X's, and those are our patients, and you can see that many of them cluster near the individual of European ancestry. Although we do have one near Colombia, and we have one near the Han Chinese South. So this was incredible to me that we can actually do this, and I think it also highlights the diversity in the public data being really useful as a control for these types of analyses, and I hope that additional samples will come online in the near future. The next thing that I'm going to discuss is phasing of variant data. So to phase genomic variation, we are just looking to see which chromosome the variant resides on. And I've shown a very simple schematic on the right where we are showing two chromosomes, and you can see at the top position there is an A on the chromosome on the left, and there is a G on the chromosome on the right, and then at the next position right below that we have a G on the chromosome on the left, and we have a T on the chromosome on the right. Now, actually doing this from short read data is challenging, and we often have to think of kind of innovative methods to get to this. But this is really important when you're looking at compound heterozygotes in autosomal recessive genes. Now, I've mostly studied autism throughout my career, and so I was really interested in looking at all those autism genes, and the phasing over those looked really nice. But I thought that for today, I would show you an example of a gene where autosomal recessive is kind of what causes the disease. And the example I'm using is phenylketonuria, and I'm looking at data from one of our patients over the PAH gene. So the first thing that I want you to appreciate from this is that the window I'm showing you here is 126 kilobase pairs in size. So this is a very large region. The next row below that is, or actually the top row in this diagram, is the coverage that we got from PacBio HiFi. So this is about a 20x genome. Right below that are the reads that were phased to haplotype 1, and for phasing, as I remind you, we use WhatsApp. Right below haplotype 1 are the reads that were phased to haplotype 2, and then directly below that is the phase block. So you can see this phase block is really big, and it goes across the whole gene, and actually it extends further beyond the gene, so it's a very large block, but I'm just focusing on the gene here. Then immediately below that, I'm showing you the Illumina phase block. So this is the short read data. Again, I processed this through the same analytical workflow using deep variant and WhatsApp to do the phasing, trying to keep things as consistent as possible. This is really amazing, and if you're thinking about autosomal recessive and you were really interested in perhaps compound hets in this gene, then knowing that the variant that I've highlighted on the top is on haplotype 1 and the variant that I've highlighted on the bottom is in haplotype 2 would be very important if those are the gene breaking events, for example. Now I'm going to show you some information where we looked at the sort of the statistics about this, and I do apologize, these were colored. I think they've been converted to black and white here. But basically, hopefully you get the sense of it. On the left-hand side, what I'm showing are the percent of phased variants. And what I hope you appreciate is that of the variants that you could potentially phase with PacBio, you can phase almost all of them, which is incredible. With Illumina, that's lower. And then likewise, on the right-hand side, what you can see is there are about 43 times more phase blocks detected in short-read sequencing data than in long-read sequencing data. And so if we just go back to the last slide for one moment, you can see there are many more Illumina phase blocks here, right? But actually having just one big phase block in the PacBio is more beneficial. And the reason why there are less blocks is because the blocks are bigger. And now I'm showing you on the left-hand side the average block length in base pairs. So on average, the blocks in PacBio data is about 186 kilobase pairs, as opposed to about 31 kilobase pairs in Illumina. On the right-hand side, if we look at the actual number of variants, we can see there are about 60 times more variants in a PacBio phase block than in the short-read data. So this is an incredibly important advance for the field of human genetics. To be able to do this kind of assessment right away just from the read data is amazing. Okay, now the next part I'm going to talk is about the assembly-based approaches. Because let's face it, if you have long-read sequencing data, one of the coolest things about it is that you can do de novo assemblies. So we wanted to look at this more formally. What I mean by a de novo assembly, just in case you're not aware what it is, it's basically building a genome from the read information only. We don't need a reference genome. All we need are the reads from this individual. Now, generally, from the data that we have, we can see there is a linear relationship between the coverage that you have and the number of SMRT cells that you sequence the individual on. So generally speaking, you can see that a 2 SMRT cell genome is about 20x, a 3 SMRT cell genome is about 30x, and a 4 SMRT cell genome is about 40x. And we had all this information and we wondered, what is the actual coverage we need to get a good de novo assembly? So this is data from one of our individuals that we sequenced with 4 SMRT cells. We intentionally did this so that we could look at these kind of metrics. So on the left-hand side of this diagram, what I'm showing you is a fairly basic plot of what's shown also on the right-hand side. So on the x-axis is the NG value, and on the y-axis are the megabase pairs in size. So NG50 is sort of easiest to explain with an example. So NG50 is the value at which 50% of the contigs in your genome are at the size that they're at or bigger. So for example, for the NG50 for 4 SMRT cells, you can see it's 54.4 megabase pairs, which means that 50% of that patient's assembled genome has contigs of that size or larger. And I hope what you can appreciate as you look at NG10, which would be the 10%, NG50 is 50%, NG90 is 90%. What I hope you can appreciate is there's this really large jump between 2 SMRT cells and 3 SMRT cells, where our assembly just gets much better at 3 SMRT cells. Now on the right-hand side, I'm showing you this across the distribution, right, from NG10 all the way to NG100. Again, you can see this big jump. So there's a kind of a small jump between a 1 SMRT cell and a 2 SMRT cell. There's a really big jump between 2 SMRT cells and 3 SMRT cells. But then moving from 3 to 4 actually doesn't give you that much better of an assembly. So we thought, this is great. We kind of have a metric of how many SMRT cells we want to run. And then we started talking to our colleagues, and we talked to some individuals at PacBio, just trying to see, are we kind of in the right range here? And the feedback we got was, you should try HiFi ASM. So I should just point out that I love the HiCanoe assembler. It's an amazing assembler. And I had not tried out HiFi ASM until that recommendation. So I thought, okay, I can do that, and I can do that very formally. So now I'm showing you on the left-hand side the HiCanoe that I showed you on the previous slide. It's the same plot. It's just got a fixed y-axis so that they're the same. And what was very surprising to me, that when you look on the right-hand side for HiFi ASM, is that the assemblies have much larger contigs. So it doesn't really matter which assembler you use, one SMRT cell is not going to be enough. But having 2 SMRT cells with HiFi ASM was very similar to having 3 SMRT cells with HiCanoe. So I want to stop for one moment and say that I'm not saying HiCanoe or HiFi ASM, one or the other, is better. I'm not saying that. But I am saying that this is a really exciting time, because I'm amazed at where we're at right now in the field, and I think that in the next few years this is going to get even better. But we wanted to dig into this just a little bit deeper, so we went back to all of our pan-genome data that we downloaded from the web, and then all of our data in-house. And we ran HiCanoe on all that data, and we ran HiFi ASM on all of that data. And what was really kind of cool and kind of surprising to me was that with HiCanoe, there was a really nice tracking between the NG50 and the estimated coverage. So most of our samples are European in ancestry, and you can see on the kind of bottom left-hand side of the left diagram that there's a really nice correlation there. And then as you go out to higher depth, what you start to see is that the pan-genome data, which is by and large been sequenced to a larger depth, has a sort of split by population. And that's kind of intriguing to me, and maybe there's some kind of genome architecture aspect to this as well. On the right-hand side, what you can see is that the correlation is not as strong, but there are, by and large, bigger assemblies, right? So overall, the NG50s are larger, but it's a little bit harder to predict, based on the estimated coverage, what your assembly will look like. However, we do see this kind of split still by population that we see in HiCanoe. So I think this is an interesting area, and but for the most part, we could probably say that the coverage is a pretty important factor. Now, I wanted to just show you what an assembly looks like. So this is one individual from our cohort where this is their visualization of a HiCanoe assembly and a HiFi ASM assembly. And that's pretty much how the data comes back. You have a big file that you have to, of course, it's sequenced, but you have to go through and parse and figure out from the bioinformatics. And it's just a really cool area to be working on at this moment. So the last part of the talk, I'm focusing on this syndrome called 9p- syndrome, and in particular, precision genomics in this syndrome. 9p- syndrome is very rare, and in fact, there are many phenotypes seen in individuals with this syndrome. So here is data we collected from multiple papers over the years where there has been an assessment of different phenotypes. And what I'm showing you in this table are the percentage of patients that have each phenotype in the different studies. But I don't really want you to know all of that information. I mean, you can look at it for sure, but the main thing I wanted to point out is that no matter where the deletion is, the individuals all have developmental delay, which is sort of an intriguing aspect once you see how the deletions look. Now, 9p- syndrome is typified by an individual having a deletion somewhere on the P arm of chromosome 9. We've gone through and aggregated all of the deletions that we can find from the literature that have a fairly good breakpoints based on the traditional kind of work from karyotype and array. So on the right-hand side, you can see this is the P arm of chromosome 9, and basically all you really need to get from this are the red bars are the deletions, and the blue ones are the duplications. So surprisingly, some people also have duplications along with the deletion. Now, at this point, you may be wondering, why is she talking about precision genomics and long-read sequencing for a syndrome that mostly has a deletion as the main genetic factor? Well, it's because 9p- syndrome, in about half of the cases, is also accompanied by a more complex event, and it's usually a translocation. So this is from our paper in this past year where we identified a translocation in a patient, and this is where we had a duplicated piece of chromosome 13 that it translocated onto a chromosome 9 that contained a small deletion. At the time, we used 10x genomics to solve this, but we are now using long-read sequencing. And our ultimate goal is to get to precision genomics for precision medicine, or PGPM, for 19-. And what I mean by that is that we can get precise breakpoint detection of the structural variation. We can look at the variation on the allele opposite of the deletion. We can look at assessment of variation around the genome. So a complete genomic workup. Because I don't know that we can ever get to precision medicine for genetic syndromes until we can do precision genomics. And that, I think, is going to be possible now with long-read sequencing. So I'm showing you here now PacBio HiFi results from our first 11 individuals with 9p- syndrome. And here I'm showing you the P arm of chromosome 9 and where all of those deletions died. Again, you can see that they are somewhat variable in size and location, just like in the published literature. And you're probably wondering, okay, Tychele, you said some of the patients have more complex events, right? And yes, they do. So here are 3 of the individuals where we have solved the more complex aspect of their variation. And I'm going to focus on individual 1 because it was a challenging case for us. So individual 1 came to us and they had done karyotype and array, and that had solved some of their variation. So we went about doing this long-read sequencing, and we were trying to figure out the whole variation in this individual. So now I'm going to just keep that little schematic on the top left, and I'll put a little box around the areas I'm looking at as I go. So the first thing that we did when we got back the long-read data was to actually look at the estimated copy number across 9p. And we did this using the read depth information, and what you can see is that there appears to be a large deletion, a mosaic duplication, and then the rest of 9 is diploid. Okay, that's great. It's really cool. However, we knew from the karyotype that 9 should be connected to 14 because there's a translocation, and you can see it in the karyotype. But we couldn't find the breakpoint, and so this was quite challenging for us. So we thought, we really got to look into this further. So we zoomed over near the telomere and looked at this little spike that you can see in the data. When we did that, to our great surprise, we saw there was actually a small deletion that's near the telomere. Then there's a small diploid segment right after that, and then there's the beginning of that large deletion. And so we were very curious, where is this piece that's in copy number of 2, and we can't figure out where it connects. So what did we do? We did de novo assembly, which is so cool that you can do it with long-read data. And when we did that, we were able to query the assembly and find that this piece of 9 had moved to the X chromosome. So this piece is present somewhere else in the genome, and that's why it's retained its status of being quote-unquote diploid. This was incredible to us, and it allowed us to finally figure out the whole suite of variation. So now at this point, precision genomics in 9p- syndrome is possible, and that's just what we're going to do. It's very awesome. Okay, so in conclusion, long-read sequencing is providing this all-in-one approach for PGPM. And what we've learned so far in these early days is that reference-based and assembly-based approaches are both important. Having a lot of control data in public databases to do frequency filtering is important. We have NOMAD, we have these kind of databases for short-read data. We need them now for long-read data, and I'm very excited that that will be an upcoming thing I hope for our field. The other aspect that I found really critical was that we can actually phase genomic variation in most of the genome, so it's a really comprehensive assay. For 9p- syndrome, we want to do PGPM for this, as I mentioned, precise breakpoint detection, looking at variation on the other allele and variation around the rest of the genome. Finally, we're now starting to apply PGPM for insights into autism. So recently we got a SMART grant from PacBio, and we're focusing on this family. And this family has 2 girls who have autism and an unaffected boy. Now this family is incredibly rare because autism mostly affects boys. And so we were very intrigued and we wanted to find out why they may have autism in looking at the genetics. And they have gone on a diagnostic odyssey not having any answers from previous genomic technologies. So with our new grant, what we've done is PacBio HiFi sequencing, bio-nano genome imaging, and 10x genomics. And we're just starting to go through that data now, and I hope that we'll have some answers in the very near future. So I will stop there and just thank you for attending my talk today, and I hope you enjoy the rest of the week. Thanks!